Hi, my name is Olivia Amador, and I'm a rising junior in Cs. I'll be presenting my project covering the optimization of hydrogel encasement for microtissue engineered neural network, also known as microtin fabrication. This microtin structure is meant to offer a potential alternative to current Parkinson's disease treatment. Parkinson's disease is a neurodegenerative disorder primarily affecting dopaminergic cells in the substantia nigra. This results in degenerated axons in the nigrostriatal pathway, which leads to a denervated striatum and affects circuits controlling patient movement. Nearly 1 million individuals in the US are currently affected by the disease, and approximately 90,000 more are diagnosed each year. While current standards of treatment, including oral levodopa supplements and deep brain stimulation, help to manage symptoms, none can restore circuit level functionality. To address these concerns, our lab has begun developing microtins, which consist of a hollow hydrogel column filled with an extracellular matrix-like solution and seeded with an aggregated mass of immature neurons at one end. The hydrogel column acts as a guide for the proliferating axons, providing unidirectional outgrowth. When fabricated with dopaminergic neurons, microtins act as tissue-engineered nigrostriatal pathways, or TENFPs, by reconnecting the substantia nigra pars compacta and striatum, theoretically restoring proper dopamine signaling required for motor regulation. However, TENFP axonal proliferation remains slow, and given that it's strongly dependent on the local neuronal microenvironment, this study aimed to optimize microtin outgrowth through the manipulation of the hydrogel encasement. So microtin casings were fabricated with methacrylated hyaluronic acid with varying degrees of backbone modification and solution concentration. As a proof of concept, we then constructed generalized microtins from embryonic rodent cortical neurons to assess the impact of the casing polymeric network density on axonal outgrowth. Our findings suggested that encasements made of higher degrees of methacrylation and lower solution concentrations correlated with less axon outgrowth within the microtin. And this may result from higher degrees of methacrylation, increasing the network cross-linking density, which in turn may reduce the diffusion of nutrients through the column and hinder axonal proliferation. We hope to use these findings to continue improving our existing model that could allow for the restoration for proper signaling required for motor regulation in patients with Parkinson's.